Hello, everyone. Welcome to the Moss Landing Marine Lab seminar series. I'm Amanda Kahn, and on behalf of the Invertebrate Ecology Lab today, we'd like to welcome all of you and our speaker. So before we get into the details of today, let's just talk really quickly about how this online seminar will work. All audience participants will be muted throughout the seminar. Please do not attempt to turn on your video or share your screen during the talk. There will be an opportunity for the audience to ask questions live at the end of the talk, just like a normal seminar. So once Dr. Carrington has finished her talk, you can use Zoom's raise hand feature to notify us, the host, that you'd like to ask a question. And the raise hand feature is located under the reactions tab at the bottom of your Zoom window. And you are invited to turn on your video during the questions portion of the talk. Also, if you are available and interested, please keep your time after the seminar free as well. Dr. Carrington is available to stick around for a more informal happy hour and chat following her seminar and question period until about 5:30 today. So now that we've got the logistics out of the way, I'd like to introduce Dr. Emily Carrington. She grew up in Michigan and North Carolina, where in her words, she developed a fascination with industrial assembly lines and coastal waves and currents, which is quite the mix. She got her BA in biological sciences at Cornell University. And then she carried on into the marine realm from there. So she pursued a PhD at Stanford University and a postdoc at University of British Columbia, where her research focused on the mechanical design of marine invertebrates and macroalgae, especially those on rocky shores, beginning on the shores of Monterey Bay. She was on the faculty at the University of Rhode Island, then shifted to her current position as professor of biology at the University of Washington, where she leads a marine biomechanics research group that's based in Seattle and Friday Harbor Labs. Her work draws on the fields ranging from engineering, biology, and oceanography, to develop a mechanistic understanding of how coastal organisms will fare in changing ocean climates. So with that, I am looking forward to hearing Dr. Carrington present her research here today, entitled Losing Their Lifeline, Mussel Attachment in Dynamic Coastal Environments. Dr. Carrington, welcome.
Great. Well, thank you so much, Amanda, for that wonderful introduction, and Alora in the background for helping me set up this talk. I really appreciate it. And it's such a pleasure to return to Monterey Bay for this talk. And like Amanda mentioned, I did my doctoral work at Hopkins Marine Station. So I was there for many, many years back in the late 80s and early 90s. So that was quite a while ago. Still one of my favorite places on earth. So I'm going to talk to you today about my research that I've been doing for several decades. I've been fascinated with how mussels attach. What I'm not going to be doing is talking about some of the other work that's been going on in my laboratory. It's going to be very mussel-centric today. I just wanted you to know that I also have interest. I just saw that Michael Graham just showed up. I've got some really interesting work going on right now with kelp, with some of my newer graduate students, Robin Fales and Kendall Murray, as well as some postdocs. I won't have time to talk about that, but I just wanted you to know that there's a lot of really fun work in the hopper there. So today will be mostly about mussels. And let's see if we can get this moving forward. Great. And I'm going to feature the work of many different people that have been in my lab over the years. So I guess the center graphic here says that a common theme is that we're interested in intertidal organisms, and we call that a biomechanics approach for the most part. And mostly what I will focus on today is the work of a postdoc, Moose O'Donnell, who's in my lab. He's now at San Francisco State. Laura Newcomb is currently at NOAA on the East Coast. Matt George is currently a postdoc at University of Washington. Molly Roberts next to him is a postdoc now at a NOAA lab in Milford, Connecticut. And then on the far right are two undergraduates that helped me with some recent work. One is in a graduate program at Columbia University and the other is a senior. So I would like to just give a shout out to them. The other reason I wanted to show this slide is here in the middle left corner. That's Hillary Hayford, who is one of your alums. So she finished her master's degree with John, and she currently works for a nonprofit in Seattle. Okay, so I would say very broadly, my research is in the realm of ecological physiology, and I like to think of that work as playing out at several levels of analysis. So anyone who is interested in physiology and ecology, it's sort of that interface between performance and function there. What I like to think of is for biomechanics, it simply fits this framework, but instead, we're focusing on a very specific aspect of physiology, and that is just how things work, what their mechanical strength is. And often the meat of what any biomechanicist will do is really in this function and morphology range. You can't see how do I get my pointer? That's what I want to know.
You can see your cursor.
You can see my cursor. Awesome.
Yes.
Okay, so most people that do biomechanics are down here in this form and function area. So they're interested in how the morphology of a structure affects how that organism works or its physiology. And what I do and an increasing number of biomechanicists do is we like to now think about how those organisms function in their environment. And so that now brings in the realm of ecology, because you need to know something about what the environment is and how that organism's interacting with it. So I like to do this with mussels, and I like to do this in wave-swept environments because they're mechanically very demanding habitats. So when the tide is in, there's often waves crashing on them, very physically demanding place to live. Okay, so I will focus mostly on mussels. They are one of the dominant species on temperate rocky shores. So what you see in this picture is a slide taken from the outer coast of Washington State, and mostly those are dense aggregations of bivalve mussels. Let's see, go forward. Okay, so one of the reasons I want to study mussels is that I'm motivated by their ecological importance. Mussels dominate this mid-intertidal zone, and people like Bob Payne, that you can see sitting here on this mussel bed, spent a career explaining to us how mussels can structure ecological communities. So one of the reasons that they do that is that they're able to dominate space. They're able to exclude many species, and they're also able to provide habitat for a lot of smaller organisms as well. The other reasons mussels are interesting and important to study is that they're important economically. So our lab has for many years partnered with a shellfish farm, Penn Cove Shellfish. It's on Whidbey Island, just to the east of where Friday Harbor is. And they are the oldest and largest mussel growers in the United States. And so the way they operate is they have floating rafts in the water, and from that they'll have these lines suspended down into the water column. The mussels will attach in these dense aggregations, anywhere from maybe 7 to 9 months, or maybe even a year. And then on a daily basis, they'll look at their orders and pull out the number of lines that they need to meet those orders. And they're able to go from farm to table in about 24 or 36 hours. So just here, I'm actually currently here in Seattle, and I see these delivery trucks all over town because they're delivering their product to the local restaurants. So what all of these processes, that is in terms of having both this ecological importance to dominate mussel habitats and to stay attached to these ropes, these aquaculture ropes. One of the key features of mussels is that they're able to form strong attachments. So what you're seeing here is a sped up movie of a mussel or half of a mussel shell, and it's taking the live animal is putting its foot out, pressing it against the wall. And then in that small depression, you can see that it's right there forming an attachment to the glass wall. And also in the groove of its foot, it's secreting proteins that set up and are molded into a fiber. So when the animal takes its foot away after 1 or 2 minutes, what's left behind is one of these abyssal threads. They actually come out white, and over the course of several days, they set up and become stronger and turn into these beautiful golden abyssal threads. So the animal makes many of these fibers, and collectively, this is known as abyssal attachment, and that's what secures the animals to the substrate. So one of the problems in both natural and farm populations is sometimes this attachment strength isn't strong enough, and this causes the animals to become detached. So on the left, we see just a close-up of a photo quadrat that I took, and this shows a patch formed in what used to be a very dense aggregation of mussels. So a storm came through, ripped about half of the population off, in this particular, half of this quadrat off. And so I've done this for over the course of several years, and on average, in Rhode Island at least, about 33% of the population is lost due to this type of damage from waves. And we also know from growers, because they tell me and they invite me out to look at things like this. This is a grower in Spain, and they invited us out to their farm to this raft, because what they wanted us to think about was this fall off, that problem that they were having that particular summer. So normally when they pull these ropes out, what they want is a very dense aggregation of mussels. And this is illustrating areas where there are bare spots on the lines. So this is a problem that growers have around the world, and they estimate anywhere from 20 to 35% of the population is lost annually, and that just cuts directly into their profits. So I approached this problem of fall off, wanting to see if I could use a biomechanical approach to predict when we had dislodgement and how many of the population would become dislodged. And so having sort of wearing an engineering hat, I thought of this as being basically a force balance equation. And it really involves both the biology side and the environment side. So on the biology side, we need to know something about mussel attachment strength. And so we need to know something about how they produce these biomaterials and how strong they are. And then there's a fluid dynamics problem here. We need to know something about what kind of wave forces or what kind of currents they see, and how these waves and currents can generate hydrodynamic forces on the animals. So whenever there is a hydrodynamic force that exceeds the strength of the animal's attachment, that's when they become dislodged. So just to give you a graphical explanation of how I would develop a biomechanical model, is that we can first go out to a population of mussels and measure how strong they are. And so we can get some idea of the distribution of attachment strengths in the population. There'll be some weak ones down here, some strong ones up here, and then somewhere, some sweet spot in the middle will be sort of where most of them are. So then we can think about how a wave might come along and apply some stress to the animals. And so we can calculate what that might be in force, in newtons. And so anytime you see animals that are weaker than that stress, we would expect them to fail. They'll become dislodged. And any animals that are stronger than that will be safe. And so the idea is, if a storm comes along, then you're going to see increased wave stress, and you would predict increased failure in that population. Another scenario that I hadn't thought of until I actually did these studies is that maybe the wave stress is constant, but there could be a change in this distribution of attachment strengths. And so there is a scenario where maybe they have weaker, the whole population has weaker attachment. And that is another way where you can see increased failure. So what I did then to develop this model was first use some fluid dynamics theory to predict what kinds of forces would be generated on mussels. So when they're in these aggregations like we see here, what tends to happen is flow, illustrated by these streamlines here, tends to get rerouted around the animals. It speeds up. This generates a low pressure system above the animals, and there's a relatively high pressure inside. So this pressure differential just generates a lift force, which tends to pull them off. And so here's an algebraic equation for how to calculate lift. So it's 1/2 times the density of the fluid times the speed at which it's moving. So this all is the fluid characteristics. And then it depends on the size of the animal and this coefficient of lift, which just simply describes how that fluid interacts with how the animal interacts with the fluid. So all of this has to do with the animal. And it turns out if you're like on the end of this aggregation, or maybe a solitary animal, what you would see more of is drag, but it's really governed by a very similar equation. Still has to do with how fast the fluid is moving and its density. And then it depends on the size of the animal, and in this case, a drag coefficient. So all of this is to say that we can predict forces on animals if we know something about how big they are, how they interact with the fluid. These are coefficients that we can figure out from lab studies using flow tanks, and if we can measure water velocities out in the field. So I've done that this was mostly done in Rhode Island, but I've done it in other areas. One nice little proxy for water velocity is to go out and measure wave heights. So we can deploy these bottom mounted wave height sensors. And from that, we know the wave height, and we can calculate the water velocity that would be generated by a wave that big. And then we can go out and measure the biology side. So we can drill holes through shells, insert fishing hooks, pull them off with spring scales or digital scales, and we can measure the force it takes to remove an animal. We know that bigger ones take more force to remove than smaller ones, and so we can divide that by the area, and that gives us an estimate of attachment strength. So those are the elements that went into this mathematical model to predict dislodgement. So we did that in Rhode Island now for 3 years. And that's what these black lines are, is a prediction of how many mussels would be, what fraction of the population would be dislodged in a given month of the year. And so what the black lines predict is that it should be highest in September and October. And then the gray bars represent what I actually observed. So I showed you a photo quadrat already. I took many of those over the course of 3 years, every 2 weeks. And that was important to do that with that temporal resolution so I could pinpoint when these patches formed in the beds. So I hope you can see that for the most part, they match pretty well. If you plot the predicted dislodgement against the observed, the model explains about 83% of the variation in month-to-month dislodgement. So that's pretty good. And what to me was really surprising about this study is that there wasn't much dislodgement predicted or observed here in these what we would consider the winter months. So you would think that waves are really high in January, February, and March. And in fact, they were, but we didn't see much dislodgement then. Most of it was concentrated in these particular months in September and October. So what we learned is there's actually pretty high wave stress from September all the way through March or April. And the reason why we see high dislodgement in this particular time is that's because they have such a weak attachment. And in fact, here's this seasonal cycle that we've seen in middleless, edgeless over a course of 6 years now. And we've also seen this pattern with mussels really all over the world. So we see this in Washington and in Europe as well. So they're about 2 times weaker in the summertime than they are in the winter. And so this is really why we see such strong dislodgement events in the summertime. And so the rest of my talk is really going to be centered on trying to understand why this happens. And it's kind of interesting to me. I entered all of this study really interested in fluid dynamics. And I thought if I just got better at predicting flow forces on animals, I'd understand dislodgement. And it turns out what I really need to know is more about what drives the production of these abyssal threads and how strong they are. And that is really the key to their ability to remain attached. Okay, so like I said, I've really been focused in the last 10 years, I'd say 15 years on what causes weak attachment, and particularly in the summer. And this is the focus of my work since I came to University of Washington. And most of this work has been done with our local bay mussel, Mytilus trocylus, but we've also worked with Mytilus galloprovincialis a bit. So what I'm going to walk you through is a series of studies that we've done, and they are aimed at answering these questions. Really, how does byssus production, how many abyssal threads there are, and the quality, how strong those abyssal threads are. And those are the 2 factors that go into creating a strong attachment. So how does that depend on water condition? So maybe there's something about the water, its temperature, pH, dissolved oxygen, or maybe salinity that will affect the strength of the byssus. And then I'll show you another set of studies where we explored whether it's simply the energetic state of the animal. Maybe it has nothing to do with the water. It's just that they happen to be really well fed, or maybe they're going to be reproductive, and that's driving their allocation towards producing these structural materials. So those are 2 different hypotheses for what could be driving variation in attachment strength. So let's start with this first one. We have set up a fairly fancy lab at Friday Harbor Labs. We call it the OAEL, known as the Ocean Acidification Environmental Laboratory. And in that, we are able to set up these mesocosms with very controlled conditions. So ocean acidification was a big deal at the time. So yes, definitely we were able to manipulate pH conditions, but we are also mindful that other environmental conditions matter as well. And so we built in the capability to manipulate water temperature, dissolved oxygen, salinity, and food supply. So this was a multi-user facility. It's still functioning today. And what I used it for, or people in my laboratory, was to put mussels in these buckets here, these different chambers, in different combinations of treatments. And then we'd let the animals, for the most part, we'd let the animals then produce abyssal threads. We take those abyssal threads over to my other laboratory where I have a tensile testing machine. I think Felicia Miller was able to use that. I'm not sure if she's here today. What this is a single abyssal thread held between some clamps. There are pneumatic grips, and this machine will pull them apart slowly and measure the force it takes to detach or break that thread. So from that, we can get an estimate of abyssal thread strength. And we will do this for those that were produced in different conditions. So we've done quite a few of these experiments, and what I will do is summarize them to say that we have a number of papers that suggest certain conditions will cause mussels to lose their grip. And I'll tell you that we think a lot of these will be associated with climate change. These conditions are associated with climate change. So the main factors that we found are ocean acidification and warming. We both expect those to cause weaker byssus. We have found that if we increase water temperature, and it needs to be above 18 degrees for Mytilus trocylus, that the abyssal threads they produce are 70% weaker. And we've actually isolated that effect to working on this proximal region of the thread. Turns out that this thread is a complex structure and there's different materials that go into it. So the temperature effect seems to be mostly in this region. In contrast, we've also seen that ocean acidification or low pH, and in this case, less than 7.6, will weaken the adhesive plaque. That's where they make that attachment to their substrate. And in this case, it's a slightly smaller, but still very substantial effect. They're 40% weaker if produced in low pH water. So we were interested in what some of the mechanisms that might be causing that. So we do know a little bit more about the adhesives of mussels, thanks to work by some really clever biochemists like Herb Waite and his group at UC Santa Barbara, as well as some others. So what they were able to do is to look at how that adhesive is formed within the mussel's foot. We know quite a bit about these proteins that they secrete. They're known as mussel foot proteins. And we know that they need to have a very low pH to be able to even flow with through those secretory vessels to get to the substrate. So what the animal does when it pushes its foot against a substrate is it eliminates protons and lowers the pH of the water. So in that distal depression, we have a pH of somewhere around 2. That allows these foot proteins to then flow to the substrate and make that initial adhesive contact to the substrate. Then the animal moves its foot away, seawater rushes in, which has a pH of about 8. And that now allows these crosslinks to form. So and make this material more cohesive. And that's how that adhesive is able to actually set up and become strong. So it's really this two-step process. It needs a low pH to get the proteins there in the first place, but it requires a higher pH to mature and become strong. And interestingly enough, a lot of these crosslinks involve L-dopa, and it uses oxygen. And so for that reason, we did think that dissolved oxygen might be involved in influencing the strength of the mussel attachment. So that inspired some work by Matt George. And so his idea was that maybe the level of oxygen in the water might affect how those adhesive plaques mature. So in the initial video that I showed you, they're producing these abyssal threads. They actually come out quite white, and it takes time for them to make these cross links. This is known as tanning. They literally tan up. And as they change color from white to gold, they become stronger, about 50% stronger. And again, this takes oxygen to do that. So what Matt was able to do then was to take abyssal threads that were made in normal seawater, but then incubate the adhesives in different water conditions for up to 12 days. So when they were in control seawater, normal pH, normal oxygen, you can see that they increase in strength. And it takes quite a long time, about a week, for them to get fully strong. And that's about a 2.5-fold increase in adhesive strength. If you put them in high temperature or low saline water, you pretty much get the same pattern. It might look a little different, but they're not statistically different. The real big differences that we see are when we see low pH water or low dissolved oxygen water. And so what happens is, yes, they make this adhesive, but it's never able to get the crosslinks necessary to achieve that high strength. Okay, so I think hopefully that will convince you that there's many aspects of the water conditions that can affect the quality of the abyssal threads. I haven't gone too much into the production, but that happens as well. And in particular, it's low pH and low dissolved oxygen that will make the material weak. So then I want to take a little sidetrack here and talk about this idea of energetic state. Maybe if they get lots of food and they're really well fed, then they can produce lots of byssus, and it's really good quality byssus. And maybe there's times when they're not so well fed that compromises their ability to produce byssus. And that would kind of make sense with the notion of them becoming weak at the end of the summer. We do know in most temperate areas, there's a lot of stratification happening, and there's not much food in the surface waters for filter feeders like mussels. So this is the work of Molly Roberts that she has published some of that work, most recently in functional ecology. And then there's another paper coming out that's in review that I'm pretty excited about seeing. So what I can say, the general framework that Molly used was this energetics framework. And the idea is that there's some food availability out in the water column, delicious phytoplankton out there. And so the mussel will feed our filter feeders, they take in that food, and there are different places where they can send that energy. So they might have sort of fixed metabolic costs. There might have, it might take some energy to produce byssus. We're not really sure at this point where to put the cost of byssus. And then whatever's left over is what people call scope for growth, and that can lead to tissue growth. So the idea is that we as the biologists are not maybe aware of what's going on inside this blue box, but what we can measure is this red part. We can measure thread production and we can measure tissue growth. And the idea is that depending on the strength of these arrows in the blue box, that you could see trade-offs between byssal thread production and tissue growth. So Molly's dissertation looked at this in a couple of different ways. First, she did some both field studies and lab manipulations to manipulate the food supply to see if there was a scenario where she could force the animals to produce fewer byssal threads. And I'm not going to show you all of that data, but what I can say is that there's not much evidence for energetic scope limiting byssal thread attachment. It seems that byssal thread production is a constitutive process, and that the mussels will do that even if they don't have, independent of the amount of food that they're being given. So they seem to be most responsive to their environmental conditions, perhaps currents or something like that, and they sense and respond that and will produce the byssal threads. So then the flip side of that question then is, now that we know they are going to go ahead and produce their attachment, does that actually affect their energetic scope? And so what I'm going to walk you through now are some field and modeling studies that suggest, yes, that mussels do produce these byssal threads. They're actually quite expensive, and it does come at a cost in terms of limiting their energetic scope, and we think limiting their growth. So that's the study here, and this involved actually a couple of other students. Laura Newcomb was a graduate student, a number of undergraduates, and Ken Siebens was involved with this as well. And it all starts with really a fairly clever manipulation where we took some scissors and cut the byssal threads that the mussels were making. And we would do this at different frequencies. So we set mussels up off the dock at Friday Harbor Labs, and some of them got their byssal threads cut every day and had to remake them. Some, we didn't hurt them quite so much, and we cut their byssal threads only once a week. And the others, and when I say hurt, these are like non-living tissues. So I don't think we were hurting them, we're just making them work harder. And so then either every day they had to reproduce the byssus, every week, and others, they were only cut once and left alone for the 6-week experiment. And then we measured how much they were able to grow. And so what we found is, first of all, this manipulation of cutting them daily, weekly, or never was successful in producing or forcing the animals to produce different amounts of byssus. So those that were severed daily had to put out quite a few byssal threads over the course of those 6 weeks. And those didn't produce very many at all. And so then when we went and measured the observed growth, how much tissue did they produce over the course of that experiment, Those that were not severed very often grew quite quickly, and we see a negative relationship here. So increased byssal thread production causes the mussels to grow less. So Molly came in and they used this empirical data to then develop a model. And she specifically, it was a scope for growth model. I'm not going to go into the details of that. It's definitely in this paper, all the equations that she used. But graphically, this is how it worked. In pink, what we have is the energetic intake that the mussels had. So this is related to how much phytoplankton was in the water column. And then we varied a thread production rate. And so in this case, that's the byssus, so the energetic cost going to byssus. And we know how much gonad they produced because we measured that, and we know how much somatic growth they had. And so the sum of all of these, let's see what we're then getting. Let me just back up a little. I think I got ahead of myself. What we know then is that there was a trade-off here between the overall growth in blue and the amount of energy going to the byssus. So that's really the trade-off that we're looking for. And it is substantial. It's not like this byssus line is really low down here. So what this shows, first of all, is that the energy going to byssus is quite a bit. We're able to use this modeling approach to estimate that it's about a joule per byssal thread that it takes. So I thought that was pretty satisfying to come up with that number. And we are able to then use the model. Oh, did I lose my graph? That's too bad. Okay, well, I did have a graph here that I managed to delete that shows what fraction of their total energy budget goes to producing byssus. And it does matter on what manipulation we used, but it's at the high end, it is up to 50 or 60% of their energy budget goes to producing attachment. So not a trivial amount by any means. Okay, so that's what I have to say about energetic state. I think that the food supply is not a good explanation for why mussels have weak attachment in summer, but I do think that if they need to make more byssal threads, it does come at an energetic cost. But I want to go back to this initial idea here where there's something about the water condition that affects byssal thread production and quality. So we have a bunch of lab experiments that say temperature matters, pH matters, and dissolved oxygen matters. Not so much with salinity. But I wanted to ask, like, when did they ever see these conditions? So to do that, we needed to go out to the field and where mussels were growing and measure the water conditions. So this is Laura Newcomb out at Penn Cove Mussel Farm. This is the owner of Penn Cove, Ian Jeffords, and I've had a really long and fun collaboration with Ian over the years. And I just wanted to say, it's really important that we have this partnership with academia, industry, and state and federal agencies. And I'll just tell you a few reasons why that is. One is that it was Ian's crew that helped us set up all of the telemetry that we needed to do to get the data in real time from this YSI data sonde. Yeah, we probably could have figured it out, but it was a whole lot easier with their engineering group that welded this beautiful stand together and worked out all the logistics. And it turns out that they liked partnering with us because they really wanted to have access to these data. They were quite happy that we were doing these experiments, but it turns out knowing water conditions and how much food is in the water is really helpful for them on a day-to-day basis. And then in terms of the state and federal agencies, we were doing our experiments. We realized we had only one site and we wanted to add a second one. And we were having trouble coordinating that, but it turned out that Ian had connections with the state that was so he was able to help us get access to a different site. And it was WDFW that was, I'm sorry, the DNR, Department of Natural Resources, that was interested in these water quality data as well. And so they bought us a whole second set of these YSI data sondes. So really working all together, communicating our needs over time, got us all much further than we would have been had we just been working independently. Okay, so these data sondes that Laura's deploying, they have five different probes on them. There's water temperature, dissolved oxygen, pH, chlorophyll, and I'm going to forget the last one. Dissolved oxygen, and salinity. And so we would deploy those and measure those continuously for many years. Let's see if there's something in the chat here. I'm not going to look at it. Okay. So to summarize that, what we have been able to show is that the mussels experience a whole lot of variability at these mussel rafts. So temperature over the course of the year can range from 5 to 24 degrees Celsius. Salinity can range from 0 when we have freshwater input from our local rivers up to full saline conditions, what we'd expect in the open ocean of 33. The pH can range from 4 to 9. Dissolved oxygen can be basically anoxic to being supersaturated. So these are very, very different conditions than you would see in the open ocean, or like many units larger, more variation than what you would expect. So for example, pH in the open ocean would range typically between 7 and 8. And so we're seeing whole units of variation larger than that. So what I just showed you would be data from what would be sensors near these rafts. I should have shown this one earlier. So here's a mussel raft with all these long lines. What Matt George was interested in, and he noticed, is that there's just a whole lot of mussels in this picture, right? We have all these different ropes. Each one of those ropes has several hundred mussels per meter. And he wanted to know what the water conditions would be right in and around those mussels in the interstitial spaces where they're producing the abyssal threads. So what he did was jam a sensor in, he set up these bags that mimicked that and jammed a sensor into them to see what kinds of conditions they saw. And so this is what he did find. So again, this is what they look like. This would be the kind of variation that we saw from the rafts over a course of a few days. So maybe 1 unit variation in pH. And this is just to show over a few days here that we saw a huge drop in pH coming all the way down to 4. So what this showed us is that on a very fine scale, I call this a micro scale, and I mean this like centimeter scale, water chemistry is really different adjacent to the animals compared to even just a few feet away. And then that is also very different than what we would be measuring well offshore. What this tells me is that using buoys from several meters away to predict water conditions near these animals is going to be a really difficult challenge. We need to know more about what's causing these small scale variations. So I got back to my roots to think about what's causing that. And now we get back to thinking about flow again. And because I knew, and I've had previous studies that say when you have really dense aggregations of animals, they knock down the flow velocity. And so this is just one illustration of that, where we're looking at the flow. Each one of these points represents the magnitude of flow when you have you have actually mussels in here. You can't see them, but the student put an aggregation of mussels here and measured flow at each one of these spots with a acoustic Doppler velocimeter. And so for each place that he measured that velocity, there's an arrow that represents the magnitude and direction of that flow. So you can imagine now there's just a wall of water coming across to that mussel aggregation. It gets rerouted over it, similar to the cartoon I showed you actually at the beginning of my talk. And there's essentially zero flow in the mussel aggregation, not much flow just above it, and it's just fine up here. So the main point here is that the velocity inside mussel beds can be anywhere from 1 to 10% of the free stream velocity. So when you have really stagnant water, that means there's not much flushing, and that lends itself to a problem where there could be buildup of metabolites in and amongst the mussels in those interstitial spaces. So when I think about this problem, here's that bulk flow going kind of across the mussel beds in this red area in between the mussels. That's what I call the interstitial spaces. And if it's not getting flushed readily, what could happen then is we've got respiration by the animals, then releasing more CO2, taking up more oxygen, which will cause the water chemistry to have low oxygen, low pH, all the conditions that are really bad for mussel attachment. And so when I think about that, like respiration driving all of that, we also know that respiration is really temperature sensitive. And so what this really sets up, which I think is really is interesting, is we start out thinking that ocean acidification and hypoxia are what cause weak attachment. Well, all of that could be driven by low flow and high temperature. So where we're headed next, we've done some pilot experiments to just give proof of concept for this. I'm doing okay on time. By putting aggregations of mussels, this sort of mimics what a long line of mussels would look like. You're looking at basically a barrel of mussels, but it's on end. And we put them across a flow tank here. This is a giant flow tank that we have at Friday Harbor Labs. And then we ran water past that aggregation and measured the amount of oxygen inside that aggregation relative to what was upstream. So these are just hobo dissolved oxygen loggers keeping track of oxygen. And so we measured the delta DO, the difference in oxygen, as a function of water velocity. And what this is just a diastream to show that at low flow, water doesn't pass through this aggregation, it gets rerouted around it. And so that just sets up a scenario where there's not much flushing going on in here. So what we are able to show that, yes, indeed, water velocity will affect the magnitude of the difference of dissolved oxygen with the free stream relative to what's going on in that aggregation. And so we can see quite a big difference, particularly when we have low water velocity. If we speed it up, you get enough flushing that there's not a problem. So this just is proof of concept that low flow causes reduced oxygen. So this is for a relatively small aggregation. We increased the aggregation size to being just bigger, 25 centimeters. As expected, we see an even bigger drawdown of oxygen because we've just got more biomass in there respiring. And when we do this as a function of temperature, as expected, relative to 15 degrees, if we increase temperature, there's more metabolic activity. And again, that causes a bigger deviation. So where we are now, I end with where we're headed next, is this working hypothesis that that we need to understand what's going on with the fluid dynamics. So here's a nice boundary layer profile of water coming across a mussel aggregation, and that the mussels interact with that fluid to change that profile so that they have low flow inside the mussel aggregation and relatively high flow above it. And so what I'm doing now, and we're funded by the National Science Foundation, is a collaboration with Mike Nishizaki. And what he's thinking about is how mussels will be respiring under different water conditions and different water temperatures. So they'll be introducing different amounts of oxygen into these spaces. And then Matt Reidenbach from the University of Virginia is going to be developing fluid dynamic models, which will allow us to predict water movement, not only water movement through and above these aggregations, but also the movement of scalars, in particular oxygen and pH, so that we'll be able to map out the concentration gradients of oxygen and CO2. So what we predict is that this could be a profile for a concentration gradient for oxygen. We would expect fairly high oxygen above the animals, but it will be drawn down to low oxygen here in the interstitial spaces. And one key feature, which I think makes this a really interesting problem from a fluid dynamic standpoint and a modeling as well, is that these are just not inanimate objects. They're mussels. Mussels are filter feeders and they have behavior. And so that's going to play into all of the fluid dynamics in these interstitial spaces as well. So specifically, we're interested in the feeding currents. We know a lot already about X currents that mussels and other bivalves can make, but there's less known about the in current, what their velocity rate, what their rates are, and where they're actually drawing the water from, because we're going to have really different predictions if they are filtering from above or if they're drawing water from the interstitial zone, increasing the flushing rate. So those are some of the things that we're moving towards doing. We just got started this summer and are just now starting to work on these behavioral studies characterizing the filtering behaviors. So what I wanted to end with is sort of more of a science communication vignette. And so every once in a while, it's probably because I work on mussels and mussels are delicious. And so sometimes our research catches the headlines because it has impacts into aquaculture and business. And so I've had the opportunity over the years, not only to talk to reporters, but to talk to politicians as well. So a couple of years ago, Jay Inslee, the governor of Washington State, came to visit Penn Cove. Ian Jeffords was there to talk about and I were there to talk about our research. So Ian was talking about the importance of Penn Cove to Washington State. It's one of those industries in a rural area providing really good wages for people. And so it's important to keep those businesses going. And then I was talking about the susceptibility to this business potentially to climate change. And so this is a great picture of me being the typical scientist, and I'm super comfortable. See me, I'm even smiling because what I'm doing is rattling off what my geeky datason can measure. So I'm saying, yeah, it measures temperature, salinity, pH, and temperature, and then I'm saying, well, that's really important because we now know from the lab that that's going to make them weaker. And so what I learned from this experience is Jay Inslee, first of all, he's really tall, so I had to look up like that. But the other thing is he was a really good listener. And he said, yes, I understand that. And very succinctly repeated what I said, maybe even better. So that told me he had read his literature on the way there. So he's like, I get it, Emily. I understand. You're going to have low pH. Low pH makes the abyssal threads weaker, and so they'll have weaker attachment. That's great, but here's what I need to know. I need to go to Olympia. That's the state capital of Washington. I need to go to Olympia and talk to lawmakers and convince them that there's going to be a problem with climate change, so that they can be motivated to want to enact new legislation to reduce carbon production, right? So that's exactly when any researcher, we start to sink because we're like, oh, I'm not as comfortable with that. And so I sat there for about 3 seconds thinking, wow, that's not my usual question. I'm less comfortable talking about implications of my research. Much rather talk about the research itself. But I quickly realized, you know what, we need to suck it up. And as researchers, we owe it to the public and we owe it to our lawmakers to give our best answer at that time. And it's okay if down the road we come up with a different answer because we come up with new information and we modify it. That's the scientific process. But on any given day, we should step up and give your best answer for that day. And so this is my best answer for today. I think, yes, we should worry. We should worry about where our oceans will be in the next 100 years. I particularly am very worried about ocean warming, and I'm very worried about local scale effects of warming on dissolved oxygen and pH. I think that mussels will become weaker and they will die. And the consequence of this for growers is that their profits are going to decline. And really my take home message here is that think about who you are talking to. I hope you are talking to maybe politicians, maybe reporters, maybe your neighbors. Take the time to listen to their questions. They're often going to be uncomfortable questions. It's my experience that they're always really good questions and maybe questions that we as scientists aren't asking ourselves. And over the years, I've found that those kinds of questions have really pushed my research into really interesting and important directions. So I'd like to stop there and take any questions that you might have. And I think I can stop my sharing even.
Thank you so
I
much.
can see people.
That was really, really interesting. And it looks like we already have a question from Andrew de Vogelaar. So Andrew will unmute you and you can turn on your video. For everyone else, just a reminder, while he does that, you can raise your hand using the reactions tab down on your Zoom screen. Yeah, looks like Andrew's ready to go.
Thanks, Emily. That was really interesting. I want to go back to your first couple of slides. When I see the mussels on the ropes, when they're dropping off in 30 or 20%, it's probably not from waves. It might be from something else, biofouling. What is your thought on how the folks in aquaculture are losing so much off of their ropes?
Yeah, so the 33% number is one that I measured for wild populations. So I'm pretty confident that's waves. And it's hurricanes, those early season storms coming in and they catch them when they have a weak attachment. The thing in Spain was really interesting. If you zoom in, which I don't think you can in that photo, but I happened to be there and we were just talking and looking and talking. And I finally said, wow, there's a lot of sea anemones on there. And they're like, yeah, it's really weird this year. There's this huge settlement of sea anemones. And so I just thought, and I didn't think much about it, but like on the boat ride back, I thought about it more and I'm like, well, they're cnidarians and they sting. And it was a lot of them there, like 50% cover or something on the mussels. So my collaborator had followed up over maybe the next year or so, and he was able to demonstrate that the sea anemones were stinging the foot of the mussels. So when they put their foot out to make abyssal threads, they would retract it because they didn't want to get stung. So in that case, I think it was a biofouling case. My work, but they're not always around. And so other places, including Penn Cove, they have about 20% sloughing each year from weak attachment. And I actually think what's happening is that the water's warming up and they get the water chemistry in the interstices is bad and it weakens the attachment.
Thank you.
All right, thanks. Next question is from Jackson Hecky. Jackson, you should be able to unmute.
Hi, fantastic talk. I kind of have a very similar question, which is, you mentioned that some of these fouling organisms, especially the cnidarians, can sting the foot, but can there be significant drag from the heavy fouling, especially in bays, if they just become absolutely like infested?
Yes, and so John Whitman had a study a long time ago with kelp growing on mussels, and then the kelp would be and a lot of I'll just back up, is that a lot of this was motivated back in the day with Bob Payne, right? I showed a picture of him sitting on his mussel bed, and really classic papers talking about patch formation, and like a lot of what we know about disturbance theory was developed in mussel beds, and it was Bob Payne and Sullivan thinking about patches forming. And so that stimulated many people, including me, trying to understand what causes a patch to form. And so it's not just waves going over individual mussels. If there's fouling on them, I think this is what you're getting at, there can be things growing on them that will promote a faster rate of patch formation than you might otherwise think. The population I worked in Rhode Island didn't have a lot of fouling on top, but I agree that that can be a problem. Yeah, here in Washington State, we have tons of barnacles, right? They're almost like, the barnacles make them like almost twice the size that they are normally. So I think that can increase drag and increase the rates of dislodgement.
Cool, thank you.
Mm-hmm.
Great question. Next question is from Travis. Travis, feel free to unmute and turn on your video if you like.
Sure, why not? Hi, Emily. Thank you very much for the talk. So my question, and I'm hoping I didn't just miss it on the graph, but going back to the abyssal thread strength experiments, looking at how significant low DO was, and then looking at the really impressive range of dissolved oxygen that you see at your study site, have you looked at how those supersaturated DO events could possibly lead to stronger abyssal thread attachments?
Yeah, so thanks for asking that. So we have done a few more experiments than what I led on, and Matt was looking at the dynamics of that. So when, I guess the key point is that we know it's not, for the most part, it's not constant low dissolved oxygen, is quite dynamic, right? So you can go from hypoxic to supersaturated if you suddenly get a big current and brings in some fresh water. And so he was interested in the time course of the abyssus cross-linking. Is it reversible? And how long does it take? And so the answer with oxygen is that if you put the threads in high oxygen, then they will set up, but that process is reversible. So then low oxygen can come in and over the course of several days make them weak. Whereas if you do that with pH, let them get strong, but then it's locked in. So if coming in with an episode of low pH will not make them weak. So I don't know about supersaturated oxygen, but I do know that this dynamics of going, high oxygen, low oxygen, all of that could be, and the timing of it, right? The duration and timing of these episodes really could be the thing that drives this pattern.
Yeah, interesting. Yeah, that was one of my first thoughts, was how they could maybe take advantage of those high DO events, right? To try and get a stronger, recruitment event or a stronger cohort. But yeah, no, that's, very interesting. That range is really impressive.
Yeah, well, I guess they don't like making abyssal threads at all in bad water. And so my working hypothesis is like bad water happens, it comes in and they clam up because they're bivalves, and that they're maybe opportunistic of making abyssal threads when the conditions are right. And then crossing their fingers that the water stays good for them to set up and stay strong. And they just have a tough time with that in the summer is my hypothesis.
Yeah, well, certainly sounds like it. Thank you.
Yeah.
All right, let's take one more question as a formal seminar before we break into a more informal happy hour, and that is from John Geller. So John, feel free to unmute.
Hey, Emily.
Hey, good to see you.
Yeah, good to see you. So I'm going to sneak in two questions. One is something I've always wondered about, and that is the strength with which the abyssal thread is attached into the tissue of the mussel as opposed to on the rock, and how that might be a part of this dynamic of tenacity. And that certainly seems like, so the thread, the non-living thread must insert somehow into the living tissue of the mussel, and that could be supremely sensitive to physiological condition. So and I have no clue how what the structure of that is, and so what do you know about that?
Yeah, so I don't have the slide of that because the animal is still attached, but I did have kind of a gruesome picture of the animal kind of dissected away out in the field to show the abyssal threads and how it was attached into there. So that attachment's known as the root. And so each thread is attached to a stem, single stem, and that stem is connected to the root, and that's what goes into the tissue. The animal can actively control that, so they can let go. So there is some process called jettisoning, and I don't understand it. I don't know that anybody understands it well, but they can just plain give it up, and then the whole kit and caboodle, because once the root is gone, then the stem and all of the threads that they have is let go. And it seems like juveniles do this more often because juveniles are pretty mobile. I've seen them in aquaria, those little guys, and you can see them like Spidey Man cruising around the tank. And sometimes they'll leave single threads behind as they're crawling because they have to get rid of the old threads, right? Or else they're stuck. And sometimes they jettison, they leave them all. And I guess that means they're only hanging on with mucus attachment with their foot.
In some cases, when I've tugged on a mussel, it's that tissue that fails rather than the thread or the attachment to the rock. So when you measure that in the lab, does that happen sometimes, or is generally that's really not the weak link in the chain there?
So when I was in Bamfield, actually I've pulled thousands of mussels off rocks.
I'm sure.
And I always would record on my spreadsheet R for root failure, versus I think I would say thread failure or substrate failure were my things, because I was also curious if it was the rock itself failing versus the threads. And root failures were rare.
Okay.
I also can tell you for Middleist Californianus back in the day at Bamfield, one of my fun facts, I guess I was collecting byssus or something, but only with that species, if you tug on the root and then just like put it on the table and walk away and come back a minute later, you can pull it right out. So they've got some response to being jiggled or tugged on that encourages, they want to jettison it. And my last bit of information is that I have seen out in the field often just byssus sitting there with the root. And so you could interpret that as a jettisoned byssus, but it's also probably what's left if a sea star ate it, or if it just died naturally. So I can't really make sense of that.
My last question relates to your speculation at the very end that changing environmental conditions could increase mortality of mussels in the wild and in culture. So as thinking genetically, I immediately asked what sort of genetic variation might there be for susceptibility to those things, and maybe there is some hope in a selective breeding sort of thing, or just natural selection in the field.
Yeah, so we are working on that now.
Great.
So what, like the holy grail would be to find the strong byssus gene, wouldn't it?
Well,
Right.
I mean, there's a bunch of proteins you had on the chart there, so those are good candidates.
Exactly. So, so that would be the idea. So right now we've got some seed money from NOAA and the Pacific Shellfish Growers Association to, I guess what we're going to do is put them in conditions for strong versus weak attachment and see if we can look to find any genes that are differentially expressed. And then we can look for individual variation, but we're just baby steps at this point. Yeah, I think that's a great question. The other thing is, if we were able to come up with a breeding program for, say, increased byssal thread production, right? You just select for that. I think what Molly's experiments show is that what you're going to get is reduced growth rate. So that will come at a cost of taking longer to get to market size, whatever you define as market size.
Better than dead, I guess.
Yeah, yeah. I think that the market is very, like they can get used to whatever size people have, right? So the French really like small mussels. The Belgians like big mussels. I think I'm not so concerned about the market reaction to that.
Thanks for a really great talk, Emily.
Thank you.
Yes,
It's fun to see, John, it's fun to see the MFPs having some, some functional use, right? I mean, and they found it.
Besides a taxonomic marker, yeah.
Yeah, they found a ton of them now. So it's getting harder and harder to figure out what any one of them does, which is really fun though.
cool. Well, thank you. Thank you so much, everyone, for the questions. I just want to give a quick moment to say a formal thank you before we go into the informal chat, but please, everyone, feel free to stick around. If you have more questions or want more conversation time, and let's give a round of applause for Dr. Carrington. Feel free to turn on your videos, stick around.